Good morning, myself, Dr.Punam Dugaje. It's cell biology lecture in the T.Y.B.Sc cell biology 5th chapter lysosomes. Its subtopic is the lysosomes origin, occurrence, morphology, and its function. So, see, first is what is mean by lysosomes? Lysosomes is a digestive. Soma means the body. It's a digestive body. So, in by Christian Belgium biologist, he discovered that the name by lysosomes, and they contain. He said that they contain digestive enzymes capable of the lysis or the digestion. So he eventually recorded in the Nobel Prize also in physiological and medical Nobel Prize in 1974. So he discovered that lysosomes concept. So see the occurring and origin. Occurrence and origin of lysosomes. The lysosomes may originated directly from the endoplasmic reticulum or from the Golgi membrane, and they associated with the vesicles formed by pinocytosis or the phagocytosis. So there is evidence that the lysosomes are also originated from the Golgi complex. So the protein granules synthesized by the ribosomes are seen in the enlargement of endoplasmic reticulum. When these are budded off, gives rise to bodies having properties of lysosomes. So they are also found by pinocytic vesicles. So got it? Is associated with vesicles formed by pinocytosis or the phagocytosis. So lysosomes have been found in both animal and plant and also in the protozoans. So lysosomes have found both in animal and plant cells and in protozoans. In bacteria, there are no lysosomes, but in bacteria there are no lysosomes, but so-called periplasmic space found between plasma membrane and the cell wall. In the bacteria, there are no lysosomes, but so-called periplasmic space found between plasma membrane and the cell wall may play a role similar to that of the lysosomes. So, its a structure. What is a lysosomes and its structures? Lysosomes have no characteristic shape or structures. So it is difficult to identify the lysosomes under electron microscope because they have no characteristic shape or structures. But they usually appear as a dense globular body and vary in the size from zero point 0.2 to zero point 0.8 micron in diameter, but they may be large in mammalian kidney up to 5 micron and exceedingly large in the phagocytosis. The lysosomes is essentially a pair of single lipoprotein. Single lipoproteins. So its membrane hydrolytic enzymes. Hydrolytic enzymes which are involved in the mechanism of cellular digestion. So if the enzymes are released, they can digest the cell. Only if the enzymes are released, they can digest the cell, and the membrane is impermeable to substrate. So the membrane is impermeable to substrate of the enzymes contained in the lysosomes. So the substrate having molecular weight higher than 200 do not diffuse through the membrane, and these indicate that lysosomal hydrolysis have no direct access to cellular component, which prevent uncontrolled digestion of the cell content. So thus lysosomal membrane is resistant to the enzymes. What is function? The lysosomal membrane is resistant to the enzymes that is enclosed an entire process of digestion. So it's carried out within a lysosome. So enzymes have destructive effect on the rest of cells. Hence the stability of the membrane of great importance to the normal function of the cell. So its function is equilibrium or the balancing. So in this diagram, there are some food particles is absorbed by endocytosis, and it forms become a food vacuoles. Then it food vacuoles it become lysosomes. In lysosomal bodies is absorb these phagocytosis or endocytotic methods. Then its some enzymes contribute its function as catalyzed, and then it becomes digestive vacuoles. And some unwanted it's eliminated by the exocytosis. So it's a type also here. So the identification of lysosomes is possible with the use of electron microscope and various cytochemical techniques. The most wide used procedure in the Gomori stain for the acid phosphatase. Cytochemical reaction for beta glucuronidase, aryl sulfatase, N-acetyls, beta glucosamidase, and 5-bromo-4-chloro chloroauridoacetase esterase can also stain the lysosomes. So certain substances like neural treat or anti-malarial drugs, vitamin A are taken by the lysosomes and may be used in their identification. So here is some enzymes we discussed. So main topic of these lysosomes, they need some enzymes also. So here see types of lysosomes. Another next point is the type of enzyme. First of all, see this diagram. Here endoplasmic reticulum. Here completely one cell and some endoplasmic reticulum, cell membrane, cytoplasm, Golgi apparatus, lysosomes. So the so evidence that lysosomes are also originated from the Golgi complex. See in this your Golgi complex is vesicle, the droplet, and they become form lysosomes. Pink in shape Golgi apparatus. Here the lysosomes may originated directly from endoplasmic reticulum or from the Golgi membrane. And they associated with the vesicle formed by pinocytosis or the phagocytosis. See in this diagram, Golgi apparatus here membrane, and then they associated with vesicle formed pinocytosis or phagocytosis. There is evidence that the lysosomes are also originated from Golgi apparatus. Got it? So next type of lysosomes. Here primary lysosomes. Second is the digestive vacuoles, then residual bodies and autophagic vacuoles. First is the first is primary lysosomes. In the primary lysosomes, here first type of lysosomes, their primary lysosomes is also called as storage granules in triangular shape. This show in this diagram is the triangular in shape primary lysosomes or storage granules is labeled. Then autophagic vacuoles, that is the digested mitochondria, auto self-eating cell, self-eating cell, autophagic. Then digestive vacuoles, after complete digestion, then forms it becomes digestive vacuoles. Then unwanted material is excreted by residual body or detection or exocytosis of the waste. So first of all, what is lysosomes? Lysosomes is so vesicles, and these the lysosomes are polymorphic. What is meant by polymorphic? Polymorphic is occurring in several different forms. It's not fixed. Okay, it's a polymorphic. Polymorphic means occurring in several different forms. So they show irregularities in size and internal structure. So because of polymorphic nature, the lysosomes are extremely dynamic, and these can be recognized after isolation of the lysosomes. When lysosomes are within the cell, they are surrounded by multivesicular bodies. These are surrounded by multivesicular bodies, smooth and coated vesicles and dense bodies. So these bodies shows fusion and fission event. These bodies event fusion and fission event. So at the present, four type of lysosomes are recognized, of which only the first is the primary lysosomes and other three may be grouped together as a secondary lysosomes. But first is the main. First is the first primary lysosomes, and another remaining three lysosomes is the group, that is the second lysosomes. Here see second one is the digestive vacuoles and residual bodies and autophagic vacuoles. Primary lysosomes. In the primary lysosomes, they are also called as storage granules. Primary is the storage granules. Each primary lysosomes is small body and its enzymatic content is synthesized by the ribosomes and accumulated in the endoplasmic reticulum. So from this, the enzyme penetrated into Golgi region, in which the first acid phosphatase reaction takes place. When the accumulated in the endoplasmic reticulum. So, from the enzymes penetrated into the Golgi region, in which the first acid is phosphatase. Acid means the first acid phosphatase is the enzymes. Acid phosphatase reaction takes place, and the primary lysosomes may be charged preferentially with one type of enzyme or the other. Got it? In the primary lysosomes, here the first reaction is acid phosphatase reaction takes place, and second one is the digestive vacuoles. Done. See in this diagram, after the food particles taken in by endocytosis, that by phagosomes, it become phagosomes, and these phagosomes are some primary lysosomes absorb in the digestive vacuoles, and it forms the autophagic vacuoles or some residual bodies. Uh, Showing this diagram by arrow. Please see carefully. So digestive vacuoles, they are also called as heterophagosomes. Hetero. Hetero means a different. Is the heterophagosomes. They are formed by phagocytosis or pinocytosis of foreign material by the cell, and these are secondary lysosomes which contain the engulfed material within the membrane and show positive acid acid phosphatase reaction. So in this, here the positive acid phosphatase reaction, and this may be due to fusion with primary lysosomes. They contain enzymes and the materials to be digested or already digested material are present. Already digested, that means the primary lysosomes or the storage granules. So here also some enzymatic reaction, that is the acid phosphatase reaction takes place. That's why. So this may be due to fusion with primary lysosomes and they contain enzymes and the material to be digested or already digested material are present. And second one is the third one is the residual bodies. Residual bodies, that is the unwanted residues. In the after digestion, the end product pass through the lysosomal membrane and are incorporated into the cell. The undigested material is left, or if the digestion is incomplete, the secondary lysosomes forms are called residual bodies. In some cells, such as amoeba or other protozoa, these residual bodies are eliminated, but in the other cells, they remain for long and may load the cell. In unicellular, the residual bodies excreted, but in our, for example, in our bodies, the cells, they remain for long, may load the cells. The residual bodies, got it? After digestion, the end product pass through the lysosomal membrane and are incorporated into the cell is the residual bodies. And another one is the autophagic vacuole. Auto means the self, automatically self-phagic vacuoles. These secondary lysosomes are also called cytolysosomes or the autophagosomes. They are special type of lysosomes in which they contain a part of the cell in the process of digestion. For example, a mitochondria or the portion of endoplasmic reticulum is a part of the cell is digested. So lysosomes are regularly engulfed by uh, xylosol, which is degraded by a mechanism called the micro microautophagy. So during starvation, the liver cell shows numerous autophagic vacuoles. During the starvation, when you do not eat some the starvation condition, the liver cell shows numerous autophagic vacuoles, and this is mechanism by which the cell can achieve the degradation of its own constituent, which have undergoes irreparable damage. Irreparable damage is the autophagic vacuoles. Mostly it found in the liver at the starvation condition. Got it? Here the type of lysosomes. There are the basic two types, primary lysosomes, and the remaining three is secondary. But in the four different types, primary lysosomes, digestive vacuoles, residual bodies, or the autophagic vacuoles. So see these in intracellular digestion there are need some enzymes. So see these enzymes, lysosomal enzymes, or its natural substrate or the source of lysosomes. So lysosomes contain variety of enzymes. Up to the present time, about 40 enzymes have been isolated. Some of the enzymes found in the lysosomes are shown in these tables. Most of the enzymes function better under acidic condition. That is the optimum pH is the 5 pH, that is the mostly acidic pH, and they are collectively called the acid hydrolysis. Collectively it's called known as the acid hydrolysis. So here in the acidic condition these enzymes is activated. So first enzymes is nuclease. Here nucleus, the natural substrate is the RNA, and its source is also source of lysosomes is many tissue of animal and plant cells. Here the nucleus. Then acid ribonuclease or the acid deoxyribonuclease. Here DNA natural substrate is the DNA and it's also source of the many tissue of the animal and plant cells. Then next enzymes is the phosphatase. In the phosphatase is natural substrate is most phosphomonoesterase and the phosphodiesterase. So it's a source also many tissue of animal and plant cells. It's common in all our body. So nuclease, acid ribonuclease, acid deoxyribonuclease, then phosphatase, oligonucleotidase, and acid phosphodiesterase is common in our tissue. It's all sources the many tissue of animal as well as plant cells. So next one is the enzymes is protease. Protease is catalyzed or natural substrate is the protein and the source of the lysosomes animals. Then cathepsin. In cathepsin, the natural substrate is the collagen. Source of the lysosomes is bone. Then collagenase. The collagenase or peptidase. Collagenase peptidase is the peptides. Natural substrate is the peptides and source of lysosomes is the animal and plants. Then next enzymes is the lipase, esterase, phospholipase. In the lipase, natural substrate is fatty acids, esterase, or the esterase enzyme esterase, natural substrate is esterase, and phospholipase, the natural substrate is phospholipids. And here also the source of lysosomes in all animal and plants. And next enzymes is some glycosidase. Here glycosidase, there are the different type of glycosidase. Beta galactosidase. In the beta galactosidase natural substrate is galactosides. All here all enzyme source of lysosomes is liver. Then alpha glycosides, glycogen. Beta glucuronidase, here the natural substrate is glycogen. mucopolysaccharides and polysaccharides. So source of the lysosomes is the liver. Then lysozymes. In the lysozymes, the enzyme lysozymes, natural substrate is bacterial cell wall and kidney mucopolysaccharides. So source of the lysosomes is egg white saliva. Sorry. Then enzymes hyaluronidase. In the hyaluronidase, natural substrate is the hyaluronic acid and chondroitin sulfate. In the chondroitin sulfate, their source of the lysosome is liver. In aryl sulfatase, enzyme aryl sulfatase is organic sulfatase. So natural substrate is organic sulfatase and source of the lysosomes is liver and plants. So see, the all important enzymes in our body or the all animals body. So this natural substrate are the source of the lysosome. Mostly in the liver, glucosides, some glucosides they are beta and alpha, beta galactosides and alpha glucosides, beta glucuronidase. Natural substrate is the galactosides or glycogen, mucopolysaccharide, polysaccharides. Or source of the lysosomes is the liver. So here the lysosomal enzymes, but function here some function of the lysosomes. Here the main important function is the intracellular digestion. So the intracellular digestion, it is the digestion which occurs inside the cell. Intracellular digestion means the internal digestion, it is the digestion occurs inside the cell. The lysosomes contain enzymes or digested material. So see, the contain enzymes or digested material intracellular. So intracellular digestion is of two types. That is the autophagy or heterophagy. Autophagy and see autophagy and heterophagy. Heterophagy, that is the phagosome, that is secondary lysosomes. Got it? The phagosomes are the secondary lysosomes. Is heterophagy and the autophagy is the autophagy. So, intracellular digestion, there are the two types of digestions. Autophagy is the intracellular digestion in which enzymes of the cell digest the piece of cytoplasmic material from the cell itself. That is, that is the digestion of endogenous material. So, example, mitochondria are constantly removed by this process, and thus there is renovation and turnover of cellular components. This is the autophagy. And the heterophagy means the intake of exogenous material into the cell and its breakdown by the lysosomal enzymes. In the endocytosis, it's phenomenon in which bulk of exogenous material is taken inside the cell, and it includes two phenomena: pinocytosis, that is the incorporation of the liquid material, that is the pinocytosis, and phagocytosis, that is intake of the solid material. So it's the liquid as well as solid material digested. So it heterophagy means intake of exogenous material into the cell and its breakdown by the lysosomal enzymes. So endocytosis is phenomenon in which bulk of exogenous material is taken inside the cell. It includes two phenomena, pinocytosis or the phagocytosis, that is the liquid as well as the solid material. Then extracellular digestion, that is the outside the cells. Extracellular, the lysosomal enzymes are also released outside the cell and hydrolyze the extracellular material. So lysosomal enzymes also involved in bone erosions. Such process may occur in rheumatoid arthritis, a human disease in which the cartilage of joints may be eroded by lysosomes. Lysosome enzymes also involved in bone erosion. Such process may occur in rheumatoid arthritis and human disease in which the cartilage of joint may be eroded by lysosomes. The dangerous extracellular digestion. Then third one is the hormonal secretion, the lysosomal protease activities, the secretion of thyroid hormone in the follicles. That is the thyroid stimulating hormone. Always THS hormone number is increases, so it becomes hypothyroidism. So hormone secretion, the lysosomal protease active the secretion of thyroid hormone in the follicles. So the thyroid stimulating hormones is the TSH. Is continuously increase. Then it become clinical significance is the hypothyroidism. Then fourth one is the lysosomes are important in germ cells and the fertilization. The lysosomes are important. So acrosomes of the spermatozoa is nothing but special lysosomes which contain protease and hyaluronidase. So these enzymes are useful for penetration of sperm into the eggs and also digestion of stored reserved material. Lysosomes are important in the germ cells and fertilization. How is the acrosome of the spermatozoa is nothing but special lysosomes which contain protease and hyaluronidase, and these enzymes are useful for penetration of sperm into egg and also digestion of stored reserved material. The next one is the role in development process. So role in development process, many developmental process involve in the shading or remolding of tissues, which the removal of whole cells and extracellular materials. For example, the degradation of the tadpole tail is produced by the action of cathepsin. Cathepsin is that is the proteolytic enzymes contain the lysosomes. Degradation of tadpole tail. When tadpole, what is tadpole? That is the frog larva is a tadpole, and the degradation of the tadpole tail is produced the action of cathepsin. It contain the lysosomes. Sorry. The lysosomes involved in human disease and syndromes. How? See, the lysosomes involved in the human diseases and syndromes. Here, in certain pathological conditions such as rheumatoid arthritis, silicosis, silicosis, and the asbestosis, that is a disease produced by the inhalation of silica or as- asbestos particles, and gout in which crystal urea accumulated in the joint, in which the crystal of the urea accumulated in the joint. There is a release of lysosomal enzymes from the macrophagus and acute inflammation of the tissue that may lead to an increase in the collagen synthesis, that is the fibrosis, and the acute release of lysosomes enzymes occurs in state of anoxia, acidosis, shock and result in increased amount of enzymes in the blood. So the lipid soluble vitamins, here lipid soluble vitamins, vitamin A, K, D and E are the lipid soluble vitamins, and the steroid hormones make the membrane more susceptible to rupture. On the other hand, corrosions, hydrocorticoids and other drugs have inflammatory action, tends to stabilize the lysosomal membrane. So here the lysosomes involved in the human disease and some are the syndromes. So next one is the storage disease. Storage disease caused by mutation that affect the lysosomal enzymes. Here the several congenital diseases have been found in which there is the accumulation within the cell of the substances such as glycogen or various glycolipids, and these are called storage diseases and are produced by mutation that affect one of the lysosomic enzymes involved in the catabolism of certain substances. For example, in glycogenosis type 2, the liver and the muscle are filled with glycogen within the membrane bound organelles. So, in this disease, alpha glycosidase, the enzymes that degrades glycogen to glucose, is absent. In this disease, the alpha glucosides, the enzymes that degrade glycogen to glucose, is absent. There are about 20 diseases due to the lack of certain lysosome enzymes. So, storage disease caused by mutation and that affect the lysosomal enzymes. Then last one is the lysosomes in plant cells and their role in the seed germination. In the plant, the lysosomes are found in plant cells in seedling. They are involved in the hydrolysis and is remove of protein and the starch during germination. When the it involved in the hydrolysis and is remove of protein and starch during the germination. So here the function of lysosomes first is the intracellular digestion. Here intracellular digestion there are the two concepts in the autophagy and heterophagy. In the autophagy here the enzymes of cell the piece of the cytoplasmic material from the cell itself, the digestion endogenous material itself, cell digestion is the autophagy. Then another one concept in intracellular digestion is the heterophagy. Here the intake exogenous here exogenous material, that is the outside the cell material. Here autophagy means the inside the material, that is the endogenous material in autophagy, and exophagy is here heterophagy is here the exogenous material into the cell by the phagocytosis or the chenocytosis. Then second one function is the extracellular digestion, is the outside the cell digestion. Or the third one is the hormone secretion, in the secretion of thyroid hormone in the follicles. If protease activate, here the lysosomes activate the protease, the secretion of thyroid hormones in the follicles. Then fourth one is the lysosomes are important in germ cells and the fertilization. And fifth one is the role in development process. Then lysosomes involved in human disease and the syndromes. Then storage disease caused by mutation that affect the lysosomal enzymes. And last one is the lysosomes in plant cells and their role in the seed germination. So what it is the completely lysosomes. Once again, we revise that all in lysosome chapter. Here the lysosomes. Lysome means the digestive, soma means the body. Digestive body in the cell is the lysosomes. Here first discovered Christian D. Dior in 1955. He stated that they contain digestive enzymes capable of lysis or digestion. So, occurs mostly originated directly the endoplasmic reticulum or from the Golgi membrane, and they associate with the vesicle forms by pinocytosis or the phagocytosis. So, lysosomes have been found both in animal and plant and in also protozoans. So here structure is not definite structure and no characteristic shape of structure. It's varying in size 0.2 to 0.8 micron in diameter. They may be large in mammalian kidney, near about 5 micron, and extremely large in the phagocytosis. Here type of lysosomes, there are the basic two types, primary lysosomes and secondary, but remaining three also types. First primary lysosomes, second was digestive vacuoles, then residual bodies, and the autophagic vacuoles is the endogenous digestion. Um, See here some lysosomal enzymes, it's important. Then function of lysosomes, we have the intracellular digestion, extracellular hormonal secretion, and lysosomes are important in germ cells and fertilization, role in development process, and the lysosomes involved in human disease and syndromes, storage disease caused by mutation that affect lysosomal enzymes, and lysosomes in plant cells and their role in cell germination. So here the frequently asked this question in exams. Describe the structure and function of lysosomes. And second question is the what are the lysosomes? Describe origin, structure, and functional significance. Then explain how to The lysosomes are suicide bags in the cell. Explain it. The lysosomes are suicide bag in the cell. Then define lysosomes. Describe their function. Is commonly asked in our semester exam. So, got it. Thank you. And next lecture on mitochondria.